Welcome grade 11 learners to another installment of Life Sciences. We're looking at comparing invertebrates today. If you guys recall, last week we spent some time looking at the different invertebrates. Today in this installment we're going to spend some time comparing the structures of different invertebrates. So let's get straight into the lesson. I hope you guys have your textbooks open. I hope you guys are eager to get involved. Let's get some discussion going on and in some interaction. So guys, we're looking at invertebrates today, but more specifically we're comparing their structures. So here I've got an overview of the lesson. We're looking at the various classifications of your vertebrates and your invertebrates, but what we're specifically looking at today is we're looking at all the invertebrates which would form this area of the phylogenetic tree. So we're looking at all these organisms on the upper side of this phylogenetic tree. And if you guys recollect, we've got classification of organisms based on the presence of a notochord or a spinal cord and a backbone, and those that don't. So we refer to that as those organisms being vertebrates, and then those that do not have them as being invertebrates. So essentially we're going to be focusing on the invertebrates. So let's go on to the next slide and let's look at this. So we have a very beautiful slide here, and that's an overall classification of organisms and especially of animals. And if we recollect from last week, we said that these are broadly classified into two groups, and of which we're going to be specializing at comparing the invertebrates today. And the characteristic of these organisms, as a recap, is that they lack a distinct backbone or spinal cord. And if we look at this illustration, guys, it shows a phylogenetic tree where you've got the organisms divided or classified based according to characteristics. So I'm going to go through some of those characteristics in a little while, but I hope that you guys can recollect from last week, and maybe this could be a discussion point for you guys before we get straight into it. So the important terminology that we need to look at today are the following. So we need to tease the words taxonomy, phylum, symmetrical, cephalization, ectoderm, coelom. So I want you guys very quickly to open your textbooks, get into a group, and have a discussion around these terms. Chat to each other and come up with some kind of explanation towards these terms. And then I'll see you guys in 2 minutes' time. Welcome back, guys. I'm sure you guys have had a good discussion. So let's spend some time quickly going through these terms, and then we'll get on to business of the day. So guys, looking at the term taxonomy, what does taxonomy mean? So remember, taxonomy refers to a classification system. So when we study taxonomy, we're looking at how organisms are classified. So basically, we're looking at a classification system based on characteristics that organisms share. And we know that we've looked at the various groups last week, And that was basically a taxonomical classification. So classifying organisms based on certain characteristics would be taxonomy. The next term that you had to discuss was, and I hope you guys came up with some good discussion around this, was the term or the word phylum. And guys, remember that we're looking at different phyla today, but that would refer to a smaller group, a large group of classification under which organisms, so it's a large group under which organisms with similar characteristics are classified. So it's a large group of classification of organisms with similar characteristics, and I'm going to try and fit that in here. Similar characteristics are classified into. And then the term symmetrical, which we've discussed last week, and we referred to symmetry as the way in which an organism can be cut and which creates identical halves or mirror images of them. And we looked at symmetrical, asymmetrical, and then bilaterally symmetrical, which we're going to discuss later on today. The other term that we need to look at is cephalization, and we discussed the significance of this, but recollect guys, cephalization refers to the concentration of sensory cells towards the anterior end. And we saw that as organisms become more developed or more specialized, we saw the development of a distinct area of cephalization, something very important to be able to recognize as we go through comparing the invertebrates. And finally, we looked at the terms ectoderm and endoderm. And these terms ectoderm and endoderm refer to the layers that are present in the embryo. So these are the embryonic layers, and if you guys recollect, it's the ectoderm, endoderm, and the outer and the middle mesoderm. So we're referring to the embryonic, and these are layers. And these embryonic layers are the layers that differentiate and become specialized as the organism develops. And coelom, guys, if you recollect again, coelom refers to the body cavity, and the body cavity is essentially the cavity that forms during the embryonic development of the layers, and that is how organisms can be differentiated based on the presence of a coelom, the absence of one, or them being acoelomic. So cool. So let's get straight on to the business of the day. So guys, very quick overview. So the key concepts that we need to discuss is that we are looking at invertebrates today, and these are animals that lack a backbone. And according to their backbone and some of the key features that we've discussed already, these organisms can be classified accordingly. And the 5 characteristics that we're looking at today in the comparisons would be their symmetry, their type of gut, the cavity that they have in terms of whether they're coelomic, acoelomic, do they show segmentation, and the development of cephalization as a characteristic of specialization. So we also discussed the concept around organisms having a body plan, and that essentially refers to the way an organism is structured. And essentially that is what we're going to be basing our comparisons today between the different groups of invertebrates. So let's look at these. So guys, what we're going to focus on today is comparing the characteristics of the various body plans of the invertebrate phylum. And the first group that we're going to look at is Porifera. And guys, I've got some beautiful images here, and you would have seen these watching some channels, some discovery channels. You would have noticed that here you've got these microorganisms that look very colorful but often seem plant-like. And these are your simple invertebrates, which are animal or organisms that are animal-like, that have these features. So what we're going to focus on today is looking at these, classifying them, looking at an example of them, discussing their symmetry, the type of tissue layers that they have, and whether they have a coelom or not, the type of gut, the skeleton that they have, and some additional facts around them. So I'll try and keep this brief because I really want to get into some questions where we can take these characteristics and interpret them in a question. Right, so the first group that we are looking at are your Porifera. An example of that is sponges. And guys, lots of you probably would have watched SpongeBob SquarePants, and this is when we refer to sponges, we essentially, SpongeBob is part of the phylum Porifera, and we're referring to the examples of all sponges. So when we look at the symmetry, these are asymmetrical. If we go back to this diagram, guys, you can clearly see that if you were to divide any of these organisms along a plane, you'll find that it does not produce a line of symmetry. So if I decide to choose to draw a line, so I'm going to change my pen, I'm going to go back there, let me just see if I can do this with a different color. So if I decide to draw a line across this section of it, guys, that does not divide the organism into mirror images, and hence we regard that as an organism having an asymmetrical pattern. They do not show any tissue layer development, no distinct body cavity, and hence we refer to them as acoelomic. And they have a very simple digestive system. It has a blind gut, and hence they filter feed. Essentially what happens is that they take in water through a common opening, which is used both to ingest food as well as to ingest the waste products. So we refer to that as a very primitive blind gut system. Okay, and let's look at the structure of the skeleton. So when we look at their skeleton structure, it is a fibrous skeleton, guys, very simple skeleton made up with spaces between the fibers that absorb water so that they can then get in their nutrients from the water. So a very primitive, a very simple method of support, as well as a method of ingestion of food. And some additional facts that I thought you guys would be keen on is that these often, they are sessile. And if you recollect from last week and previous episodes, we've discussed that sessile refers to organisms that are found attached to a substrate and do not move for most of their life. And hence, they rely on the movement of the currents in the ocean to bring food towards them. And that is how they have quite a simplified mechanism around which they survive and are able to establish themselves in their environments. So that was the first group. Let's look at the second group. So the second group of phyla that we're looking at is phylum Cnidaria. And guys, you would have seen beautiful images of these. Here we've got the jellyfish and the sea anemone. And often I know learners struggle with pronouncing sea anemone. And if you've watched documentaries like, or movies or programs like Finding Nemo, you will see that Nemo was obviously well embedded and protected in a sea anemone. So cool, let's look at this. Again, the key features we're discussing are the symmetry it shows, the tissue type, the coelom, the gut system, the type of skeleton, and some additional facts. So when we look at the phylum Cnidaria, we think of examples of jellyfish, corals. So these you would have seen, guys. These often lie on the beds of the oceans, often attached to rocks. Beautiful. Some of them often move, and hence you find that here's a transition from being sessile in the previous phylum to organisms now that are both sedentary, sessile, and have the ability to be able to move. And now we're seeing the progression of movement, a sense of becoming more specialized. So let's look at it. Do they show symmetry? Yes. So they are radially symmetrical. And guys, if you recollect from last week, we said that when we look at radial symmetry, if we were to cut up this organism along the radius, you'll notice that it will produce mirror images, multiple mirror images of the left and right hand sides. And hence we refer to the Cnidarians as being radially symmetrical. Cool. They show a tissue layer development being diplobastic. In the previous phylum, we noticed that there was no tissue development. Here we find 2 layers of development from an embryonic development stage. The absence of a coelom, so no coelomic cavity as such. And what do they have? They have a blind gut. That means they're a common opening through which they ingest food and they release their waste. Okay. However, what is unique to them is that they have the presence of tentacles, and you would have seen that in this diagram, the presence of tentacles here around the oral opening or the anterior opening. And these tentacles have sting cells called nematocysts, something that is characteristic of the phylum Cnidaria. And these nematocysts essentially are sting cells that release a sting, that trap and immobilize the organisms. And now we're seeing a more advanced form of nutrition where this organism is able to release a nematocyst, sting its prey, and then ingest it and draw them in with the tentacles. So quite an advanced mode of nutrition compared to Porifera, which was much more sessile. It waited for food to come to it. Here we're seeing these organisms having the ability to be able to attack or even pull food towards their oral opening. So again, we see the development and specialization becoming more complex. They lack a skeletal system, guys, which again is something that we see synonymous with the first group. And most of these are marine species. We find them predominantly found in a marine environment. So guys, we've covered 2 of these phyla. I'm going to do another one before we go into a break. But I think you guys should engage with this at a level of comparing them with me. So again, we're looking at the next phyla, and the next phyla is called Platyelementus. And guys, the word platy Platyelementus refers to flat worms. So here we're looking at these worms that have a flat or dorso-ventrally flattened. So remember last week we referred to the terms the dorsal surface and the ventral surface. Now we're looking at organisms that have had their body surface flattened to appear flat-like. So essentially what we're looking at are the flatworms in this particular phylum. So Platyelementus, and examples of these will be the Bilharzia worm as well as the tapeworm. And guys, you've seen images of tapeworm. It doesn't appear round like the earthworm. It's a flattened body surface. It's especially well developed for its mode of nutrition. Often they are parasitic, they live on other organisms, and they rely on their flat body surface to be able to absorb nutrients from their host. So like these again are bilaterally symmetrical. Again guys, we're seeing a specialization from the previous group of being radially symmetrical to these being bilaterally symmetrical. Let's look at what that means. That means that if we draw a line across the middle plane, the sagittal plane, we're going to get a left and right mirror image of these organisms. Again, we could do that here as well. If we draw a line along the middle, you'll find that it produces a left and right mirror image that resemble exactly the other half. So again, we see a form of specialization which we see being carried through to the next phyla. So the first was asymmetrical. The second phylum that we looked at previous to this was radially symmetrical, and now we're seeing the development of organisms becoming bilaterally symmetrical. And notice, this is going to be a feature that carries on and into the next phyla. So I would remember, if you recollect, bilaterally symmetrical is seen the development of a proximal end and a distal end. So the development of a head and a tail. And for the first time here we see the development of a distinct head and a posterior end. And that we know, for a head to develop, here you've got sensory organs, and these sensory organs means that a mouth, a head, sensory organs show the process of cephalization, or the development of an anterior end being a more advanced center for neural activity and coordination. These organisms show 3 layers of development from the embryo, and hence we refer to them as being triploblastic. Again, we see the absence of a coelom, a blind gut, not as evolved as the next phyla, but again, so they have a gut system that has a common oral opening and an opening that is used for the digestion of waste products. Again, organisms lacking a skeleton, so they rely on their body being filled with tissue fluid that gives them shape and structure. Some additional facts, and I did mention this towards, many members of this group are internal parasites in other organisms. So liver fluke we see is a free living organism, but the tapeworms, the Bilharzia worms are all examples of parasites that live within a host, and due to their mode of nutrition, they can absorb their nutrients through their body surfaces. They have appendages that allow them to attach onto the host, and often they have a mouth part with hooks that attach them onto the intestinal lining of an organism. So a little more evolved method of nutrition, where they're now living as parasites and being able to absorb nutrients through their body surface as well as through their oral openings. Cool. I think guys, we've covered 3 phyla. We're going to have a short break, and when we get back after the break, we're going to continue comparing them, and we're going to take some questions. Guys, welcome back. I know that you've had a chance to relax, unwind. Let's get on with this. We need to still compare the invertebrates. We've gone on to the 4th phylum. We've spent some time looking at cnidarians. We looked at your flatworms, Platyelementus. Now we're looking at your roundworms, or the Annelids. And guys, if you look around the country, we've been having lots of rain. And if you do go out into your garden, you'll probably see that you've got lots of these earthworms bringing up some of the soil. And look around, dig a few holes, move some stones, move some soil, you'll find that you're probably going to find an earthworm in your garden. So the best way is go out there, guys, go into your school ground, find a flatworm, find a roundworm. Okay, so let's look at Annelids. Again guys, beautiful images of flatworms. Another example of a flatworm would be a leech. Again, a leech is a parasite. It's an ectoparasite, so it attaches onto a surface of organisms and it absorbs, and it will suck up the blood from the organism. Okay, so we're looking at some of these can be parasites, but not necessarily. Okay, so the examples in Annelida are we looking at leech and earthworms. Again guys, as I said, these would show bilateral symmetry. The tissue layers, so these are, I'm just going to erase this, these are triploblastic guys, so they have 3 layers, triploblastic, so they show 3 layers of development. They definitely have a coelom, they have a through gut, and they have a hydrostatic skeleton. Now we're seeing the development of a through gut. What do we mean by that? Again guys, a recollection from last week. A through gut refers to a digestive system in which, if you look at the diagram guys, you'll find that we have a distinct anterior opening and food enters, or the organism will ingest through that, and as the food goes through the rest of the body, it goes through a process of digestion until which it produces the fecal pellets at the posterior end. So we have a distinct anterior opening and a posterior end. So this type of digestive system is referred to as a through gut. So now we see the invent of a through gut system. Again, these organisms show a form of a hydrostatic skeleton. Again, body tubes filled with water that give the organism shape and structure. Something that we see synonymous with all your flatworms, with all your leech, with all your annelids. Cool. Some key features. What is good to note about this is that these organisms have a segmented body. They are found in soil, they could be marine and freshwater environments, and the word annelid literally means ringed body. So their body is divided into segments, and if you do look at this diagram guys, you can clearly see that here we're seeing a segmented body. Even if we go into this and look at the leech, if you look carefully, you'll see that there is details around how the body is divided into these segments, and you can clearly see here the segments pointing out a segmented nature characteristic of the group Annelids. So guys, we're going to go on to the last group and probably the most diverse group of the invertebrates, and this is the group which we all often dread in terms of examples, but these are probably my favorite group, and these are the Arthropoda. And guys, the term Arthropoda refers to organisms that have jointed appendages or jointed legs. So the largest group in the invertebrates are the arthropods. Very diverse group. There are 4 or 5 subgroups in which these are further classified, but we're going to have a very big overview of arthropods, and then we're going to get into some questions. So some of your creepy friends are here, and these are the spiders, and here we've got an image of a crab. So you've got flies, spiders, crabs. Most of the insects that we see all belong to the group Arthropoda. These are bilaterally symmetrical, and again we see this characteristic being carried on. Triploblastic, having 3 germ layers from an embryonic stage. They have a distinct coelom, a body cavity, a through gut system, so an oral opening. We see definitely the process of cephalization, or where we see a distinct development of nerves or sensory, as well as in spiders, the development of an anterior end having a concentration of nerve rings and a nerve and sensory organs. So we see the development of cephalization amongst these organisms. What type of skeleton do they show? They show an exoskeleton, guys. And this is gradually shed in organisms in order for the organism to grow. And we refer to that process as molting. And this is one of the challenges that arthropods have. Because they carry an exoskeleton, that exoskeleton needs to be often molted and being replaced with a new outer exoskeleton. And so these are part of the challenges that an arthropod would have. And so we'll need to discuss that as a development strategy that arthropods have, and the advantages of such a skeleton and possibly the disadvantages of it as well. So cool. So guys, I'm going to get into some questions, but what I'm going to do is I'm going to go through 2 or 3 of these, and I'm going to give you a few minutes, probably 2 minutes, or maybe a minute, I think, to work through 2 of these, and then we get back and we'll discuss these. So let's look at these multiple choice. 1.1. An animal that can be cut in only one place to produce 2 identical halves is A. Asymmetrical, radially symmetrical, C. Bilaterally symmetrical, D. None. Let's look at another question. 1.2. A skeleton that consists of fluid inside the animal's body is A. Exoskeleton, B. Endoskeleton, C. Hydrostatic skeleton, D. None. So guys, 2 multiple choice. I'm sure you guys can have a quick discussion. I'll give you guys a minute and we'll get back after a minute and we'll look at these answers. So guys, let's look at these answers. I'm sure you had a good discussion with your colleagues. 1.1. An animal that can be cut in only one place to produce two identical halves. So guys, remember the key words, two identical halves. So for an organism to be cut in one place and to produce two identical halves, we refer to that as being bilaterally symmetrical. And the link to bilateral should be the word two, and hence we know that along that one single plane, it divides the organism into two identical halves. So well done to those of you that got it right. The others, guys, let's hope we can get the next one correct. 1.2. A skeleton that consists of fluid inside the animal's body is referred to as what? So guys, remember we looked at endoskeletons, exoskeletons, and obviously hydrostatic skeletons. We know that organisms with an external outer covering, and we see this in arthropods, have an exoskeleton. We and other vertebrates have an endoskeleton. So humans have an endoskeleton, and that's found internally. But we also did see that those organisms, like the tapeworm, they have a body structure that is given shape with body tubes that are filled with water, and the term hydrostatic skeleton. And remember that the word hydro refers to something that is fluid-like, and hence the answer for that would be a hydrostatic skeleton. So well done to you guys if you got both correct. So we've got a few more, but I think I'm going to work with you through these here so we can have so you can have a discussion along with your friends while we work through this. Remember, read the question, pick up the keywords so that we can focus on the correct option in the answers. 1.3. Name the structure which are stinging cells that paralyze prey in Hydra. So guys, remember we spoke of the second group in Cnidaria being characterized by the presence of nematocysts. So these are the sting cells that they will kill, and they'll release a toxin that will immobilize their prey, and with their tentacles they tend to bring the food source or their prey towards the oral opening. So guys, so what is the answer? So is it a through gut? No guys, that's not correct. Is it the nematocyst? Correct. Or is it the mesoglia? No, this is a body and tentacles. No. So the correct answer here would be the nematocyst or the nematoblast, which we see characteristic of the group Cnidarians, and the example that we referred to here are Hydra. So guys, next one. 1.4. Animals that have two layers in the embryo are called. So guys, remember the word two again, so it should refer to the word di. So we know that organisms with a two layer in the embryo are referred to as being diploblastic, and the word triploblastic would be organisms that have three layers. Polyblastic I haven't heard of. Monoblastic would be something that has a single layer, and hence the correct answer here would be diploblastic, and we saw that as being characteristics of the first and second groups of organisms that we were comparing. And finally guys, let's look at the last one before we take some longer questions. The type of polysaccharide that forms an exoskeleton in arthropods is called. So guys, if we look at most of our arthropods, if you look at your insects, if you look at crabs, if you look at, they have an external body covering that is hard, and it's not bone structure, but it's of a different nature. It's made up of a polysaccharide, and not the conventional polysaccharide, but this polysaccharide gives it a hard external tough protection. So guys, remember that we know that plants have cellulose, and hence it's a plant, that's a plant-like polysaccharide. Glycogen is a polysaccharide that's found in animal cells, so that's incorrect. And we've got two that we need to decide between: pectin and the word chitin. So the answer here is chitin, and pectin would be a type of polypeptide that's found in some of the other organisms. The answer here we're looking at is organisms that have skeleton. Arthropods have a chitinous exoskeleton. So well done to those of you that got the answers right, and those of you that didn't, I think it's a good point of discussion that you need to go back and chat to your friends, open some books, do a bit of research, and try and establish why you've got your answers wrong. It's possibly maybe the terminology that you've misunderstood, or possibly you haven't interpreted the question correctly. So look out for hints in the question, and often, as you'd seen, I looked for the words bi, diploid, and I linked that to finding the correct answer. So these are the key concepts that you need to have when working through these multiple choice questions. So guys, we're going to go into the next section. Here we're going to look at some longer questions. I'm going to go through a few, and then I'm going to possibly pause and give you some time for you to have a discussion with your friends. So let's read a few of these, and then we're going to work through them. So question 2.1. Why is a blood system unnecessary in either free-living or parasitic worms? So quite an open question. A blood system unnecessary in either free-living or parasitic worms. So essentially we're referring to, in this group of organisms, we're referring to the tapeworm. We're referring to the tapeworm, and guys, if you recollect, the tapeworm is an organism that has a large surface area, and that is made about by the flattened segments that it has, and it essentially needs to absorb nutrients through its body surface. So the question is, why is a blood system unnecessary? Why is it not needed by free-living parasitic worms? So essentially, if we look at more evolved, more developed organisms, a blood system is required for the transfer of nutrients from it's required for the transfer of nutrients from the which is being absorbed by the digestive system, and then to the rest of the cells in the body. However, if we look at the parasitic worms, these organisms rely on absorbing the nutrients in a parasitic way from their host. So they have a large surface area, and their surface and their body surfaces are thin, which means that each of the cells absorb and get the nutrients purely from the absorption that occurs through their cell walls, and that is essentially why your parasitic worms lack a developed blood system. Okay, so cool. So I'm going to go through this here, and possibly going to give you guys a little break so that when we get back, you guys can work through some of these with me. So I'm going to read this question. I want you guys to think about this question over the break. When we get back, we're going to have a further discussion around this. So 2.2, what is a through gut? That's the first part of the question. And the second part is, discuss the advantage of a through gut in comparison to a blind gut. Quite a nice question to look at. So guys, I think you guys deserve a break. Think about this question over your break, and when we get back after the break, we're going to try and answer this question. Guys, I hope you had a good discussion about that question. So we're going into the last segment. We're going to work through a few more questions, and I want you guys to have a discussion amongst yourselves. Chat to your teacher, open those books, and maybe we can come up with some good answers to some of these questions. So before we went into the break, we looked at this question, and I needed to come back to this question and discuss the answer. So what is a through gut? First part of the question. And the second part is, discuss the advantage of a through gut in comparison to a blind gut. So I'm going to go on to, I've got some space here to work through this. So guys, let's see. So again, so we've got to discuss what is a through gut. So a through gut is a digestive system which has an anterior and a posterior opening. And what that essentially means is that food now enters from one end and it goes through to the other end where it can be digested. So we have the ingestion of food and we have the egestion of food. So essentially what we're saying is that here we have an anterior end where food enters and it travels through, it undergoes the process of digestion, and then whatever is undigested is digested and removed from the body. So why is this an advantage to the organism? So we've got to discuss the advantage of this in comparison to a blind gut. So the first part of the question is, what is a through gut? So we've described that a through gut is a system in which it has an anterior and a posterior opening through which ingestion occurs at one end, and the food and the waste is then ingested through the other posterior end. Now we've got to discuss the advantage of such a system in comparison to a blind gut. Guys, if we compare a blind gut, and I'll try and draw a diagram of a hydra here, and a blind gut essentially is an organism that has a common oral opening through which food will, let me just change my color of my pen. So here you're going to find food that enters, goes into the body cavity, undergoes digestion, and then through that same opening the waste products are released. So here we have ingestion occurring through the anterior end and the process of egestion occurring also through the same anterior opening. So if we compare the advancement of a body structure of a feature of this organism, so would you be at an advantage if you had a common opening through ingest food and through to ingest food? Definitely not. It will mean that the process of ingesting food and releasing food needs to be well timed. However, in organisms that have a through gut system, they can constantly ingest food. The food undergoes a process of digestion, and then it goes through an opening at the end where the process of digestion can occur constantly without having to interrupt the process of ingesting food. So quite a nice question for 4 marks. Let's move on to the next question, folks. Okay, so I'm going to get to the next question, which was discuss the ecological role of earthworms. So guys, and you would have seen, so this takes us to a process called vermiculture, and vermiculture is quite it's okay. Let me describe this process. So here we're using worms to decompose organic material, and they break down the organic material, and in that process they digest and they return the organic nutrients back into the soil. So over and apart from the organic material being decomposed, earthworms also burrow into the ground, and that means they create air pockets, air spaces in the soil, and this helps in both drainage of water, as well as for creating aeration, and this helps in preventing the decomposition of roots due to the collection of water. So drainage of water, so it creates these drainage holes because they tend to burrow into the soil, and it allows for aeration of soil. That simply means that it creates air spaces through which seeds can germinate so that the nutrients can get in, so that water can be absorbed into the soil. And finally, they also play a significant role in the decomposition and the breakdown of dead leaves. So we refer to that as organic matter. So if you think of all your leaves in your garden, that can be taken and put into a heap. If you had a host of worms, an example earthworms, these earthworms covered with soil will eventually break down the leaves and will return the nutrients from the vegetable matter, from the leaves, back into the soil, thus creating very fertile and rich soil that can be used in an from an ecological perspective in culturing and growing and increasing the fertility of the soil. Cool. So quite a nice question that looks at the significance of earthworms from an ecological perspective. So we're going to go on to the next question, and let's see. Okay, cool. Okay, I'm just going to get to this question quickly. What are the main advantages of an exoskeleton? So we've got to discuss the main advantages of an exoskeleton. That's the first part of the question. And then we've got to discuss these disadvantages, discuss how disadvantages have been overcome in arthropods. So, okay, so what do we mean by, okay, I'm just going to get to this question. For some reason my pen's jumping now. Okay, cool. So let me not press the screen again. So what are the main advantages of having an exoskeleton? So guys, remember that an exoskeleton is an external protective covering. So when you look at arthropods, they live in environmental conditions that subject them to being threatened by changes in the environment. So it offers protection. And remember that protection from other organisms, it prevents them from dehydration. So it allows them to be able to conserve the loss of water. We have skin, and we know that skin can easily dehydrate in the harsh conditions. So it reduces the possibility of water loss. It also offers protection. So protection, and it is lightweight. It is light in weight, and this means that it makes the organism more buoyant. It makes them more aerodynamic, allows them to be able to have a less body mass, so that they're able to spend less energy on the process of movement or even flying. So in comparison to their weight, the weight of their skeleton makes them significantly lighter, and thus enabling them to be able to move effectively in their environment. The second part of the question is discuss how these disadvantages have been overcome. So the main disadvantage of an exoskeleton, so we've looked at the advantages which I just discussed. It's also important for us to analyze this question, and the question basically has got the part where we're looking at the disadvantages. So what do we mean by the disadvantages? What is the negatives of having an exoskeleton? These organisms need to molt, and you know that a molt is the process of shredding the outer external covering, and during that process, when the organism has to molt, it is an energy expensive process. So it's expensive from an energy point of view, energy expensive, but it is meaning that when this organism has to molt, it makes the organism probably vulnerable, so it increases their possibility of becoming, so they're energy expensive and they become vulnerable to danger because they lack their outer covering, and hence this increases their risk of being attacked or being destroyed or eaten up by other stronger. Okay, cool. So I think we've worked on this question. I've got a nice activity which I'm going to work with. I'm going to get through some of these, and I hope that we can do as much as we can. So this is an image of a phylogenetic tree, and a phylogenetic tree basically shows the classification of organisms along a system where we have a common ancestor, and through the process of change and development, there is specialization, and we characterize organisms based on certain characteristics, and that is how we classify them. So here we got an example of a phylogenetic tree. So let's read the question. Study the phylogenetic tree and answer the questions that follow. So as I said, this is a representation called the phylogenetic tree. Let's see. Let's read through these questions very quickly, and then we'll try and answer each one of them. According to this phylogenetic tree, which group were the ancestors of the animal kingdom? So guys, if we go back to this diagram, all these questions can be answered by purely looking at the diagram. So if we look at this, all these organisms, guys, they share a common link to the ancestral protist. And this illustration shows you that the ancestral protist were the common ancestor that gave rise to all other invertebrates. So the answer to that question would be the ancestral protist. And guys, by that, if you go back to microorganisms, we looked at the various different microorganisms. We looked at viruses, bacteria, and then protista, which was the third group, after which we saw the development of fungi. So when we look at the group protista, we saw your plant-like and animal-like protista. These are the ancestral animal-like protista that we're referring to, that gave rise to the development and specialization of your invertebrates. 3.2. How many phyla are shown in this tree? Cool. And which phyla are the phyla that have not been studied in this segment? So if we go back to this, so remember we said that the phylum refers to a small group of a classification which has many organisms with similar characteristics. If we look at this phylogenetic tree, guys, it refers to the Poriferas, which we've studied. It refers to the Cnidarians, which we've studied, Platyelementus, which we've studied. We've done the Annelids, we've done Arthropods, and so we've covered 1, 2, 3, 4, 5, but in total there are, I'm going to tick the ones that we haven't done in pink so you remember. So we haven't done nematodes, we've not done mollusks, echinoderms and chordates, but your chordates are the ones that have a vertebral column. So of this, how many in total? So we've got 1, 2, 3, 4, 5, 6, 7, 8, 9. And of the 9, we've spent time studying the 5 group. Cool. So again, the other 5 are not of significance to us at this stage. 3.3. The first major split in the animal kingdom was into radial and bilateral symmetry. So if we go back up to this diagram, it shows you that the first significant split was here, where they showed radial symmetry and bilateral symmetry. So if you look at this, here we've got, to the left of this diagram, we've got all the organisms along this branch being classified according to radial symmetry. If we go further up, and to this, on the left hand side of this branch, we're seeing these organisms all having common characteristic of being bilaterally symmetrical. So it's very important that you understand that at some point, so all these organisms show true tissue, but at this point of development, the ones that split up from these showed radial symmetry, and these further down all showed bilateral symmetry. So it's very important for you to be able to understand that a branch indicates the development of a new characteristic, and the continuation of that line indicates the continuation of that specific characteristic. So guys, let's answer this question. Which phylum does not form part of this split? So if we go back to this diagram, we're seeing here radial symmetry, bilateral symmetry, but we also see that there is a phylum that is not part of that split, and that phylum essentially, guys, is this branch here, and that takes us through the group called Porifera. So if we look at this diagram, the Poriferans are the group that have split up with a characteristic that has neither radially symmetrical and bilateral symmetrical. And if we compare this, guys, it ties in with what we first did, where we said that your Poriferas are asymmetrical, and classically here we're seeing that explanation of that in a phylogenetic tree. Let's look at this question before we go into the next. Which phylum has radial symmetry? So let's go back to this diagram. So guys, let's identify radial symmetry. Here's radial symmetry. Let's see where this takes us. So it takes us all to the group called Cnidarians. So all of our Cnidarians have radial symmetry, and that is essentially how you should be interpreting this phylogenetic tree. I'm going to take one more question, guys, before we close this segment. The second split is animals which have a coelom and animals which lack a body cavity. Which phylum has no body cavity? So let's go back to this diagram, and here we're looking at coelom and no coelom. So here we find the development of coelom. If we go down this way, these are the organisms that lack a body cavity. So guys, what we've spent this segment looking at is questions, and I hope that you guys have enjoyed, interacted with these questions. There are some questions that are on this that we haven't covered, but I'm sure that you guys can open to your textbooks, find some questions, get involved with your friends, have a discussion around these. It's very important to be able to take those key features and compare them. So from my side, guys, I hope you enjoyed the lesson. All the best, take care, work smart.